Hi friends, welcome back to the JQ Biology, and today I will be described about the southern blottings. That's why southern blotting should be carried out and what is the application of the southern blottings and why say it is called hybridization. Okay. Southern blotting is a specific technique to identify specific sequences within the plasmid or within the genomic DNA. Okay. Suppose that I am saying about the states of the southern blottings. Okay. Suppose that there is DNA sequence, okay, plasmid DNA, and so we want to know the specific sequences within this DNA. Then how we can identify? It's need to be fragmented in many parts. Okay, how we can fragment? You know that's restriction endonuclease cut into many different fragments into the specific site. Okay, suppose that we want to know that there is a specific sequences within the DNA. We want to know that there is specific sequences. If we want to identify, then we require to restriction endonuclease digestion. Okay, many restriction endonuclease is available now. Eco R1, Eco thirds, many. Okay, so this will fragment into different parts of the small DNA fragments, different parts. Okay, but even still we cannot identify that if that is, suppose that this is A, T, G, C, T. This is the sequences. The red color showing specific sequences within this genomic DNA. Okay, after restriction digestion, there is anywhere this fragment should be available, or a number of fragments of that types may be present in these sequences, but how we can identify? There is not, we can detect the specific sequences of the DNA. Okay, then the gel electrophoresis, you know, I think all you understand about the gel electrophoresis. If you not understand, then go back to the JQ Biology lectures of the gel electrophoresis. Then you can find the agar gel electrophoresis and polyacrylamide gel electrophoresis. Then on the basis of size, we can separate the DNA molecules. Even I have described certainly that this is the gel electrophoresis. Okay, agar gel electrophoresis separate the larger molecule of the DNA, but the polyacrylamide gel electrophoresis separates smaller DNA fragments. Okay, so on the basis of this, there is a many lane in this gel, and the fragments would be lowered on the agarose gel. Okay, on the basis, then this is a marker. You know this molecular markers, that is the sequence is known, specific sequence is known, is run in one well of different size of the KB. Suppose that this is 50 base pairs, 100 base pairs, 200 base pairs, and so on. Okay, you know that the separation would be takes place from negative to positive. Okay, this is the positive negative end, this is the positive ends. DNA move from negative to positive because the phosphate groups is present in the DNA, and so this move to the lower directions. The direction would be here of the DNA movements. And also on the basis of size, the larger size would be remain in that positions, or on the basis of sequence of the size, that is the smallest size would be lower, that is there is bands. Okay, this is the first lane. Second lane also can be matched here, or not match or match. Okay, third even can match from one to another, or not match from one to another. Okay, the fragments, different fragments have been separated by the gel electrophoresis, you can see here. Okay, then the smaller fragments would be run faster, and the larger fragments remain above the shorter fragments. Okay, so how we can detect that is the specific sequence is available or not? Even you can detect, even we can detect on the basis of marker, that is base pairs. Okay, but point out, this is 200 base pairs marker. You look here that the sequence may be different. But even you go to the higher, that is 300 base pairs, for example 300 base pairs, okay, 300 base pairs, then it may be similar or not. This is looking here slightly similar or may be similar in the gel, okay, by the naked eye, by the on the gel, by the UV films. You know that we cannot see the naked eye. We need ethidium bromide to mix within the gels, and the ethidium bromides, what, in the presence of UV, fluorescent would occur in the DNA and it can be detected, and there is close similarity, but we cannot say that this is the exact sequence in this gel, okay, we cannot say. This is the problems in the gel, okay. So what is needed to identify specific sequence of the DNA? What needs? Then southern blotting or southern hybridization is required. So this requires single stranded. This is the separation of the double stranded DNA. Okay, double stranded of the DNA, but requires single stranded. What needs? Alkali treatment of this gel. Alkali treatment of this gel. Okay, alkali treatment. Make single stranded. The solution of the alkali already have been made, and this gel is dipped within this alkali solutions, it becomes single stranded, okay. Then this treated gel would be transferred where? Would be transferred for the blotting, okay. What done? This is done for, suppose that this is the beaker where there is a buffer in that sides, okay. That is buffer in that sides where a paper, filter paper is put up here, and this filter paper is saturated. Point out, this filter paper is saturated, okay. And after then this gel is put up and cover or from above the filter paper put up here, okay. You know that in the gel there is pore, cross linking of the agarose gel. In the agarose gel cross linking would occur and there is pore, and you know that the concentration of the gel or concentration of the agarose determine the pore size. More the concentrated agarose gel, smaller the size, and the lower the concentration and higher would be the pore size. So there is a pore, and when the buffer, there is no problems. That buffer is absorbed by the paper, and this paper through the capillary actions, the buffers move to the gel, and above this is the gel where our fragments of the DNA is lie here, okay. Then above it we put up the nitrocellulose paper. So nitrocellulose paper, this is more important, nitrocellulose paper, okay. The same size of the nitrocellulose paper as compared to the gel, same size. Then this DNA will again goes to up directions with the buffer through the gel, okay. Moving of the buffer to the upside, there is no problems, but the DNA is attached here because a specific group is present here where the DNA binds to the nitrocellulose paper. This is nitrocellulose paper, okay. DNA when move up directions, then this DNA is attached to the nitrocellulose papers. This nitrocellulose paper is only specific to the DNA, not to the RNA. Point out, not to the RNA, only specific to the DNA. But nitrocellulose papers have many free binding sites. This is the DNA that may be black, but many free binding sites would be available for at the time of probing or hybridizations. It may maybe create problems. So this nitrocellulose paper is treated with Ficol bovine serum albumin, BSA, bovine serum albumin, polyvinyl pyridine. Okay, pyridine. This is the chemicals that will, and also irregular amount of tRNA. Why it's required irregular amount of tRNA? Because the empty sites within the nitrocellular papers will be filled up, and there is not non-specific binding will be occur of our probes, our desired probes. Okay, because this treatment will fill up the empty sites. So this chemical is required to increase the specificity of our desired fragments or desired sequence. Okay, this treatment is required. Okay, on the above nitrocellulose papers, number of towels, paper towels, number of paper towels I am making for papers in the black. Okay, to absorb the buffers, okay. So you will see here that the buffer will transfer to the nitro above, the gel to the towels, but it will be absorbed, and also there is no problem of the buffers, but DNA cannot transfer to the towel. DNA cannot transfer because there is specific groups in the nitrocellulose papers that's bind to the DNA and stop to their movings to the upsides. Okay, then this will be removed. This nitrocellulose will be removed from this. Okay, removed from this. That would be equal to the same. That would be equal to the same. Okay, so then you will see that there is nitrocellulose paper that will binds to like this, looking like this. Okay, but even we cannot know that what is our specific sequence, what is our desired sequences, that is within the genomic sequences or within plasmids. Okay, so this nitrocellulose paper is packed. Where packed? Packed into a specific type of polythene. What? Specific type of polythene that's contain probe. What is probe? Probe is a specific sequences that is known, that is artificially made, and it becomes radio labeled. Okay, and probe is known. That is, suppose that this is the probe. Okay, this is probe, and probe is radio labeled. Okay, probe is radio labeled, but points out there are a lot of radio labeled fragments is available here. Okay, this is radio labeled. In these sequences, when there is specific sequences is found, you know that in the normal conditions, natural conditions, the DNA sequences is C complementary to G, A complementary to T, double stranded forms. So the probe is also single stranded, and here after denaturations there is also single stranded. Okay, so the probe when get complementary to their sequences, then what happens? It will be hybridized. So why is it called hybridizations? It will get hybridized, and then specific hybridization, that is stringency. A word is used, stringency, that is slowly hybridization will be take place. If you hybridize a number of probes simultaneously, then it will get mismatch with the specific sequences. So in first, stringency will carry out, that is stringency, a word, carefully hybridizations, and then washing of unhybridized probes or unhybridized probes. Washing should be need. Okay, Again, after then, hybridization is required with specific sequences with 2 to 3 times. Okay. Then, if you will found that this is nitrocellulose paper that will create bands. Okay. If get hybridized specifically of that sequences, the sequence is same, all the probe sequence is same, and that will get hybridized. If this get hybridized on the presence of X-ray, on the presence of X-ray, it will turn to black. That is, photographic film is obtained, that is called autoradiography. Okay. Autoradiography. This is called autoradiography. Okay. Then the specific sequence is known, and you will find that there is our desired specific sequences is available in this nitrocellulose papers, and we can detect correctly or near about correctly that specific sequence is this, but we cannot determine on the basis of gel electrophoresis. Okay, there is smears, there may be smears, or there may be similar size looking in the presence of UV, but it may be different. But on nitrocellulose paper determine the specific sequences, specific length or specific base pairs with the help of southern hybridizations. Okay, so even we can map with the restriction fragment length polymorphism of the human genomes that differ in single nucleotides, or even around the restriction digestions or restriction sites, around the restriction sites. Okay, also we can know the specific sequences within the DNA with the help of southern hybridization, and this is the very reliable techniques. Okay, to know the specific sequences, sensitive techniques. And then nitrocellulose papers, I have described about the nitrocellulose papers that binds to the specific DNA sequences, but today now used nylon membranes. Nylon membranes. What is the benefit of nylon membranes? The benefits of the nylon membrane is that will bind to the DNA as well as also from the RNA. Okay, as well as also from the RNA, and it can be reutilized. But the nitrocellulose paper we cannot reutilize. Okay, so nylon membrane is today now used because again utilizations and bind to the RNA and both as well as DNA. So this was to know the techniques to identify the specific sequences of the DNA, and hope this lecture would be beneficial to you. And so if you like this lecture, then please click subscribe button and also share for your friends. Thank you.